Good morning, everybody. I am Rebecca Katz. I'm a professor and director of the Center for Global Health Science and Security. And welcome to our sixth of this calendar academic year, the Global Health Security Seminar Series. We do these seminars about once a month during the academic year, and always this early in the morning. So we are very appreciative for those of you who are on the East Coast who have woken up early to join us this morning. We are really excited to have our colleague, Dr. Colin Carlson, who is going to present work at the Verena Consortium, which is an interdisciplinary collaboration that's working to advance viral disease prediction and to try to strengthen health security. As a researcher at the center and the principal investigator of Verena, Colin has been working to understand the intersection of global climate change, biodiversity loss, and emerging infectious diseases, and how this relationship can be used to better inform pandemic preparedness and climate change adaptation. So thank you to Colin for joining us this morning and for in advance for sharing all of your work with us today. What we're going to do here is that Colin will speak for about 35-40 minutes, and we also have online Siobhan Robinson-Marshall, who will be monitoring the Q&A. And so please, along the way, add in your questions, add in your comments, and once Colin's done presenting, we'll have a back and forth, or as much as one can on a online webinar. So again, thank you so much for joining us this morning, and Colin, over to you.
Thanks, Rebecca, for that wonderful introduction, and thank you everyone for coming. I'm gonna throw up these slides real fast. So thank you everyone for coming and for being here at 8 a.m. 8 a.m. is not my time of day, and I'm sure it's not your favorite time of day for many of you. So it's wonderful to be doing this. My name is Colin Carlson. Like Rebecca said, I am an assistant research professor at our Center for Global Health Science and Security. I am a global change biologist by training, but I work at this intersection of climate change research, emerging disease forecasting, and health security. And I'm here today to talk about a research program that I run called the Varina Consortium, and some of the work that we've been doing to predict viral emergence. And along the way, I hope to answer a couple questions about where SARS-CoV-2 came from and what the next COVID-19 could look like. So to tell the story of Verena, I have to start at the beginning. And like all good stories, this one starts with a friendship. This is Greg Albury, a postdoc in the biology department. He and I have worked together for about 2.5 years. Greg and I are both disease ecologists, but we come at that from very different perspectives. Greg is interested in understanding animal movement and behavior and how those contribute to disease risk. Well, I tend to prefer the human side of things. I usually work in medical geography, thinking about the global distribution of infectious diseases. Sometimes I even do outbreak response. Sometimes I do pure climate work. But what Greg and I have in common is this shared passion for disease ecology and a shared interest in one specific question. We want to understand how climate change might be connected to the next pandemic. And so in 2019, we started working on a study of climate change and the global virome. What we found was shocking. Species are on the move and they're taking their viruses with them. And when they arrive in new places, that lets viruses jump into hosts for the first time. What we found was that in the next 50 years, those cross-species transmission events could happen at least 10,000 times just because of climate change. In a half century, the global virome would be completely transformed. This problem stretches far beyond how we normally think about viral emergence. And we knew that that meant there was a mountain of work left to do. This was the start, not end of the question. And so we started putting together a team. We focused on experts, but mostly early career researchers, people who want to get up and do things and try new things. We focused on bringing together specialists, people with very specific skills and specific expertise, but who were willing to talk to each other and learn from each other. And we called that the Viral Emergence Research Initiative, or for short, the Verena Consortium. Our mission is to build tools that let us predict which viruses could infect humans, which animals host them, and where they could emerge. You can think of this like a big game of Clue, but instead of Colonel Mustard with the candlestick in the conservatory, it's horseshoe bats with the SARS-like virus in the wildlife trade. And I know that there are a lot of groups out there that work on problems like these, particularly as we look to the end of the pandemic. There's a sort of renewed interest in making sure that something like COVID-19 doesn't happen again. So it's probably useful to upfront explain where we fit in that ecosystem and what differentiates our group. For one thing, we are not a nonprofit or an NGO or a government program, but we're also not a club or a professional society. We are a team of researchers who each have roles outside of Verena, but we come together to do work that we think is impactful, work that we don't think we could do in isolation. Today, there are about 30 people who've been involved in Verena at various points, and together they've written about 50 papers in various stages of publication. And we've done all that with very, very small seed grants. And if you're wondering how we can do so much with so little, I actually kind of want to start by giving away the secret recipe up front, because I think it's actually important to understand how we think about these problems, not just what the endpoints are of the work. So here's what we do. We start by building data sets together, data sets that can change the kinds of questions we can ask. And then we work together to analyze them in new ways that bring together our expertise and turn an evidence base into a hypothesis test into actionable science. The reason that this model works so well is that everyone on our team approaches problems from different backgrounds. And so they think about things slightly differently in their own work. And that helps us do new things together. For example, Greg is interested in understanding why some animals share viruses, but others don't. And he uses their ecology and their evolution to try to predict that. In contrast, Barbara Hahn's lab has spent years building tools that let us predict which animals are likely to share viruses with humans, tools that let us build a list of species we should be sampling that might be the host of Zaire Ebola virus. Then you have someone like Nardis Malentz, who's building technologies that let us look at a virus's genome and try to predict if it could infect humans. Now, all of these people think about problems differently in their own work. But what we do is we use network science to put them all in the same conceptual framework. Here's an example of a network. We know Zaire Ebola virus can infect humans, and we know that Bombilli Ebola virus can infect Angolan free-tailed bats. Nardis's research asks, can Bombilli Ebola virus infect humans? Well, Barbara's work tries to figure out where we should look for Zaire Ebola virus in nature. Even Greg's work can be framed as a network problem, where we're asking, do these two hosts share any viruses at all? All of these are subsets of a much bigger problem, which we call link prediction. This is a standard problem in computer science, where you look at a network and you try to identify unsampled links, or to put it in the terms of virology, you look at this network and you try to figure out which viruses could infect which hosts. The reality, though, is the global virome is much bigger than this toy network. In fact, it's pretty huge. This is a representation of the mammal virus network that we know about today, which only captures about 1% of the viral diversity in nature. Most of the global virome is still unsampled, and the data tells us that there's a lot missing. We also have another problem sort of built into this, which is that the data that we use to generate this network is incredibly fragmented. Different studies each capture different parts of the network. But until Verena, there hasn't been a concerted effort to synthesize all that information in one place. But that idea, data synthesis, is the core of how we approach problems. When I say data synthesis, I mean that we are going into the literature or into gray data. We're pulling out reusable data either by hand or increasingly using text mining. And we're developing data sets that change the horizons of the questions we can ask. Data sets that are so much bigger than what came before, they change the fundamental scope of the science we can do. For example, let's go back to that network. This network is generated from a data set we call Virion. This is our flagship data synthesis program, where we've pulled together the largest database of vertebrate virus associations in the world. We started that process by reconciling all those different fragmented data sets into a public product called Clover, which we released back in January. Since then, we've been building on that data set. We work with NCBI to get GenBank and the Sequence Read Archive into there and to build metagenomic data into this for the first time. As of a couple weeks ago, we are the first data set to include all of the newly publicly available data from the USAID Predict program. Our goal is to be the comprehensive endpoint, to bring everything together in one place and make it completely open without barriers to the scientific community. This project has been ongoing for a few months. We'll be releasing Virion very soon. Stay tuned for an announcement about that. But this is the process, right? We build, we refine, we expand, we synthesize. Now, there's more to data synthesis than just building new data. Like I said, data synthesis is about changing the horizons of the questions that we can actually answer. So what can we do with Virion? When we started building models together, we wanted to make sure we weren't just covering ground that other people had already worked through. So we built this library of studies that were either things we had worked on individually before this group, or that other teams had published, or that grabbed us from other disciplines. And we realized that that was actually probably a useful resource for a lot of people. Maybe you're a PhD student looking for a topic for a computational chapter. Maybe you're a WHO expert looking for the latest and greatest in zoonotic risk prediction. But whoever you are, you can go to our website as of today. This is the first day. It is not password protected. And you can check out the Hivemind Database, an open library of annotated studies on predicting the global virome. This is where we get our inspiration. And with that in mind, I want to tell you how we build on those studies. We're trying to test out new kinds of methods that could permanently change how we think about prediction. For example, let's go back to Nardis's model. So Nardis is interested in looking at the genome of a newly discovered virus, usually the first information you get, and trying to figure out, based on just that information, does this look like a virus that can infect humans? What we can do is we can take the Virion data set, we can predict the undetected parts of the network, and we can fill in the most complete network possible. Then we can take that network and we can use matrix algebra to collapse it into a set of features that are called embeddings. And embeddings are a way of talking about sort of where viruses fit in the network, what their position is. We can take those embeddings and we can add them to Nardis's model. So we say, here's the genome of this virus, and also here's sort of where it fits in the global ecosystem of mammals and their viruses. And when we do that, the predictive power of these models skyrockets. And that shouldn't be a surprise, right? If we understand the general rules of cross-species transmission, we can do a better job of understanding zoonotic risk as a special case. But this shows you exactly how much data synthesis can accelerate prediction and how it lets us do things that no one has actually tried before. We can also use data sets like these to test hypotheses about how the world is changing. For example, in our climate work, we use Greg's viral sharing model to try to project what would happen in a changing climate. We find that as the planet gets warmer, every species on Earth is moving towards the poles or to higher elevations, trying to track shifting habitats. And bats have a little bit of an advantage because they might just be able to use flight to kind of beat that race. That's great news for bats, but unfortunately it's bad news for us because every time species meet for the first time, they'll share viruses. And we think this could happen thousands of times. This isn't just a problem for those species. It's also a problem for us. There's sort of a weird artifact of human history that comes into play here. A lot of human settlements have been built in places with a stable climate at the edge of biodiversity hotspots. Think about what you learned in AP World History about the Indus River Valley civilization, right? There are reasons that human settlements exist where they do. And unfortunately, those are also the places that a lot of species are going to move to in a changing climate. And that means that's going to be where they share viruses. And as they jump between species, that might make it easier for one of those viruses to jump to humans and start the next pandemic. This isn't just a future problem, too. The world that we live in today is about a degree warmer than pre-industrial temperatures. And in our most recent analysis, we found that this probably puts us at increased risk already. So we can use this kind of work to look at a map and pinpoint where we think viral emergence risk will be highest because of climate change. And we can start planning accordingly, not just in the health security space, but as part of climate adaptation. In addition to our work on climate change, we're also very interested in urbanization and land use change. We already know that urbanization is a major risk factor for disease emergence. And we've used our data to show that animals that are capable of living in cities have a higher diversity of zoonotic pathogens than those that prefer undisturbed settings. But when we use path analysis on that model, we learn that this is actually only true because they have a higher total diversity of pathogens. This sort of rules out this idea that urban environments help zoonotic viruses emerge faster. Instead, it might mean that species that can live in cities or end up living in cities are generally more prone to disease. Think about the kinds of animals that do well in a city. Think about rats or squirrels or raccoons. But it could also be something else. It could also be an observation bias, right? We might just know more about the pathogens of species that live closer to us. And telling the difference between those two things is at the core of how we understand the relationship between urbanization and disease emergence. And so we're continuing to sort of unpack this in our work because we need to understand what the relative role of those processes is. But this is what we can do. We can use these data to not just predict how the world will change, but understand how it is already changing. And we don't stop there, right? Because we want to make everything we do actionable science. And we have a number of pieces on the way that connect our data and our modeling to real world problems. For example, we recently held a one-day workshop about zoonotic risk technology, which is what we're starting to call these models that look at a viral genome and try to predict zoonotic risk. And we've written a study, which will be on our preprint server very soon, where we explain how do these models work? Why are they getting better over time? What could they do for global health? And where might we hit new problems because that technology exists? For example, imagine that a bad actor used artificial intelligence to predict all of the spike proteins of undiscovered coronaviruses. And then they went and they tried to patent all of those sequences. That might make it harder the next time one of those viruses does emerge in humans to develop therapeutics and vaccines or distribute them effectively. But we don't know because we haven't really anticipated what the world looks like with this technology in it. Now, this is a complicated problem, and it's one that you can only pull apart when you bring together experts on machine learning and virology and law and global health practice. And there's no other group that does that as proactively as we do. Similarly, on the topic of global change, we've got a number of pieces that use evidence-based science to talk about spillover risk in the context of wildlife trade or deforestation or climate change. We also have some forthcoming guidance about something called spillback, which is when a human pathogen makes its way back into animals. Now, you've probably heard about spillback a little bit in the last few months because there's been a tremendous concern about COVID in mink and about COVID in mink farms making its way back into natural ecosystems. There's a lot of reasons to worry about that, but it turns out this sort of broader nebulous fear that this might prevent us from eradicating the virus, that it might make it easier for the virus to sort of spill over again, which we call secondary spillover. That phenomenon has actually sort of never really been seen up to this point. And when we build an evidence base to go looking for this, it turns out that spillback is chronically underdocumented, it's understudied, and we don't really have enough data to build a good framework for risk. And so in our policy guidance, we dive into why that is, and we explain where researchers can put their effort and how policymakers can approach that risk in the next COVID. That's how we approach problems. We gather and synthesize data. We try to ask new kinds of questions. We try to answer them with new methods. And then we try to translate that into some sort of impact for global health or conservation or broader issues. And with that in mind, I want to kind of transition a little bit, because I told you I was going to talk about COVID today, and I'm going to. I'm going to take a little drip of water, and then we're going to move on. There are a few really big questions about COVID that are floating around in the literature right now. And most of them are more unresolved than I think is comfortable. Where did SARS-CoV-2 actually come from? What could the next COVID look like, and how soon might we be dealing with it? And then in the context of our work, there's sort of a question within that, which is, does the work that we do actually prepare us for that next COVID? And so I want to try to answer each of those questions as sort of honestly and thoroughly as possible as we can using this approach. To do that, I want to start by rewinding all the way back to the start of last year. Right as the pandemic was kicking off, right? We knew that this was a novel coronavirus. We knew that it looked a lot like SARS, but was not SARS. There were a lot of question marks, and there are still a lot of question marks. We knew pretty quickly that SARS-CoV-2 is a virus that almost certainly originated in bats sometime in the last few decades. And we found closely related viruses in horseshoe bats, and we found closely related viruses in pangolins. But so far, there hasn't actually been a perfect match. And this is sort of to be expected, right? We know that these viruses emerge not just by normal sort of genetic evolution, but also through recombination. And that makes it kind of hard to track down how viruses originated. We also know that a lot of the viruses that look like SARS-CoV-2 still haven't been discovered. There are probably hundreds of SARS-like viruses in bats that we still don't have a name for, we still don't have a genetic sequence for, and that makes it really hard to place SARS-CoV-2 sort of in this broader picture of the origin of these viruses. And so we're working with all these surveillance gaps, we're working with all these data gaps, and we're trying to sort of get to the bottom of a narrative, right? We want to be able to say, this is the host that it started in, this is how it made its way to humans, and this is what it tells us about the future. So back at the start of last year, we started to put together a project that we thought might help fill in some of those gaps. Like we always do, we started with data synthesis. So we started by putting together a list of every known bat host of beta coronaviruses. And using those data, we tried to get a couple different places. First, we wanted to know how COVID might have gotten to humans. With coronaviruses, we're used to the idea that these viruses often jump from bats to humans through an intermediate host. In the original SARS outbreak, we saw the virus probably jump through civets. With MERS, we see MERS circulating today in camels, and it's been circulating in camels since at least 2012. These viruses seem to like to use what we call a bridge host. It makes it a little bit easier to make that jump if you're a virus that's been co-evolving with a host for a while. And so we want to know what that bridge host is, and we want to try to use our models to get as close to an answer about that as we can. So we went back to Greg's viral sharing model to see if we could get some answers. And what we did is we took the sharing model and we looked at all of the species that we think could be sharing viruses with those two horseshoe bats that have closely related viruses to SARS-CoV-2. Now, most of what they're going to share viruses with are other bats. And that makes sense, right? Because species tend to share viruses with closely evolutionarily related animals. But there's a couple other things that come out really strong in this chart. One of them is pangolins. Pangolins are the top predicted species to share with Rhinolophus affinis. The other is carnivores, and that includes civets. It also includes mink and some of the other species that we've seen SARS-CoV-2 spill back into. And it manages to make those predictions without any prior knowledge about SARS-CoV-2. All we need to know about is the host. So we can refine this even further. Let's take the top 10 predictions that come out, 5 for each of these species. This is out of 5,000 species of mammals. This is a very, very, very small set, right? This is just the top 0.1% of our predictions. And there's a couple really crazy things that jump out. First, you'll see that Malayan pangolins make the cut, and we now know that those host very similar viruses to SARS-CoV-2. Now, there's been a lot of focus on the wildlife trade as a reason that those pangolins were exposed to these viruses. They were confiscated from the wildlife trade. But what our model is looking at is viral sharing in nature. Just because of where these animals live, we actually expect that pangolins and bats are probably able to share viruses on a pretty regular basis, maybe without the help of humans. And in the time since we published this finding, it's also turned out that in Africa, there are these extensive deep burrows where pangolins and bats live together and presumably share pathogens completely naturally. So this is a pretty big win for the model, all things considered. But it also tells us something, right? It tells us that maybe these viruses made the jump into pangolins before they got to the wildlife trade. There's something else in these results that really jumps out. This species, Melogale personata, it's called the ferret badger. These animals are on the short list of species that are now suspected to be the actual bridge host of SARS-CoV-2. And that's based on a World Health Organization investigation, which has dug into Huanan market, dug into the role of wildlife farming in southern China. It's looked at human contact networks, and it's tried to come up with a list of what might plausibly have been the animal that helped these viruses make the jump. And again, it's in our top 10. So these are shockingly prescient predictions. They are certainly better than you would expect at random. And believe it or not, that's actually still just the warm up, because what we actually want to know is, where did this virus come from in the long run? What are the bat species that are out there that still have undiscovered beta coronaviruses? There are hundreds of bats in the world. And as of the time that we compiled our data set, only 79 of them are known to have these viruses, but presumably hundreds more might. So normally, we'd probably go and take our data and we would build a model to try to guess which species might have undiscovered viruses. But again, we wanted to do something that no one has tried before. And so we built an ensemble of 8 different models. Now, some of them use bat ecology to predict viral reservoirs, like Barbara Hahn's work has done in the past. Others use network science to try to make those inferences without ever bringing in external predictors. And again, this is where we get an advantage from using network science. Now, there's some broad correlation between all of these predictions, and there are some species that come out on top of those predictions no matter where you're looking. But we find that the different kinds of models make really different predictions. The trait-based models that we're used to in this field, they tell us to go look deeper in Southeast Asia. And that's helpful, right? Because that is where we are likely to find the bat that hosts the ancestor of SARS-CoV-2. But the network models point us in a different direction. The network models say, hey, there might be a whole hotspot of undiscovered beta coronaviruses in the Amazon that we just don't know about. And so this shows us how we can change the answers with network science and how we can get unexpected results that might lead to new avenues for discovery. But we didn't want to stop there. Over the last year, we have tracked all of the newly discovered beta coronaviruses in bats that have been published, put on GenBank, announced via press release. If you follow me on Twitter, you may have seen me do little threads about this. And all of this is in plain view of the public, not just on Twitter, but also in our beta coronavirus database, where we keep track of every bat that has been discovered to be a host of these viruses. Now, this isn't just a bookkeeping exercise. This lets us hold ourselves accountable for the work. We can use these data to try to actually measure how well our models do. Normally, when you do machine learning, you split your data set into training and test, and you try to convince the reader that the model has done a good job on data it's never seen before. But your training and test data are usually generated from the same sampling process. And that means that even in those circumstances, you're probably going to do better at random than random. What we have here is an opportunity to actually fully break these models and to see how they behave when you push them to the limit. So let's walk through this graph. On the x-axis, we're talking about the percentage of bats that the model thinks might have undiscovered viruses. Well, on the y-axis, this is the percentage that the model gets right from the newly discovered hosts we've been tracking. So think about it like this. If it's a coin toss, if the model is truly random, half the time it says yes, half the time it says no, it's going to come out 50% on the x-axis. It'll say half of all bats have an undiscovered beta coronavirus. And in the actual newly discovered hosts, it will be right about 50% of the time. And that's our expectation at random. That line says, if you're above this line, you're doing well. If you're below this line, you're doing bad. And we can use that to measure how well we're doing. So for example, our ensemble says, okay, there are 1,100 bats out there that we don't know have these viruses. I think about 1 in every 4 of them might have an undiscovered virus. But it gets 65% of the newly discovered hosts correct. So that means it's doing better than you would expect at random. And that validates that the exercise is useful. But it's not great accuracy. If we want to see great accuracy, what we have to do is split those predictions back into the component models. And when we do that, we get one really clear answer. The trait-based models work really well. So this tells us a couple really big things that we didn't know before we did this. First, if we want to find more viruses like SARS-CoV-2 or Ebola virus or anything that we really care about, right? We actually do really need to know about the ecology and evolution of the hosts. Basic science about bats goes a long way for this kind of research. Second, there's a huge room for improvement with these network models. This isn't, let's throw the baby out with the bathwater, these models don't work. This is, we have a process to benchmark the accuracy of these models, and now we can refine them and make them better. So we've gone from data to model to prediction to validation. And even then, we don't want to stop there. We want to make these predictions fully actionable. And so to complete the life cycle of the work, we're very excited to be partnering with the Field Museum in Chicago to test our predictions using their collections. Over the next year, we'll be going through at least about 1,000 samples with this small pilot project. And we're hopeful that we'll discover new viruses, we'll continue to test and improve our predictions, and we'll show the value of museum collections along the way. Collections are chronically underfunded and one of the most valuable institutions in science, and they matter for this kind of work. So this is a great opportunity for us to, again, bring together folks in the same space and do transformative work we couldn't do on our own. And even then, we're not done, because we're also working to target that analysis a little bit by synthesizing other kinds of data about coronavirus surveillance that are useful. For example, in this meta-analysis that we'll be publishing in the next few months, we found that there are particular tests you can do of particular samples that are more likely to pick up these viruses. And we can apply that knowledge to help optimize the Field Museum's viral discovery program. And if that wasn't enough, we're going through the whole process again in parallel with rodent hantaviruses, working with Chris Forbes's lab at the University of Arkansas. So we are putting these models to the test, and we are going looking for viruses that are relevant to health security. I'm going to pause there for a second. So our team is trying, at least, to do the whole risk assessment pipeline. We predict which animals are hosting the next pandemic virus. We go sample those viruses. We rank them based on whether we think they could infect humans and how deadly they might be. And all of that is an unprecedented step for doing risk assessment in one place. And this is the point in the talk when, if I actually had something to sell you, I'd probably tell you that means we can stop pandemics at the source. We as in a collective, but also we as in my team. But this isn't that talk. The truth is, people have been trying to sell this idea of predict and prevent for a while, like decades, and it always rings kind of hollow, particularly now that we've seen exactly how unprepared we were for COVID-19. Now, for decades, people have been saying that this is because predict and prevent is sort of just out of reach, but I think COVID tells us that's not true. Not because of how bad the specific pandemic was, but because we had more forewarning about it than we've had for maybe any previous pandemic. We did a test run of COVID 20 years ago with the first SARS outbreak. And in the decades since, researchers have been sounding the alarm over and over that we might be facing this kind of pandemic someday. Today, more than a year into this pandemic, we still don't know which bat species hosts the ancestor of SARS-CoV-2. We still don't know how it jumped to humans. Maybe it happened in a wildlife market, maybe not. Maybe it was in a pangolin or a ferret badger or an animal we haven't even guessed yet. There are still so many gaps. But what we do know is that people saw this coming. And even with all those gaps in our knowledge and all those gaps in our predictions, I can tell you confidently today, it could still happen again. What I would contend to you is that we should listen to pandemic experts who have been doing this for decades. This isn't a lesson about prevention. It's a lesson about preparedness. There will be more pandemics of zoonotic viruses. And when they happen, we'll be called on to do this kind of work again. When that comes up, collaborative models like the Verena Consortium can help us accelerate science and can change the outer limits of what we can do with the basic science that exists. I think in that framing, it's also a little bit easier to construct an argument for why our work matters to pandemic preparedness. Let's go back to this idea of viral discovery. There's 1,200 bat species in our study out of about maybe 1,400 in the world, and about 1,100 of them aren't currently known to host a beta coronavirus. Any one of those 1,100 could be the bat that hosts the ancestor of SARS-CoV-2, or the next virus like it, but we don't know today. They also host a lot more viruses than just beta coronaviruses. There are hundreds of viruses in each of these bat species, each of which could plausibly pose a threat to health security someday. Some of these will be the start of the next pandemic, but today, they're all undiscovered. What our tools let us do is narrow down the bats that might be hosting undiscovered beta coronaviruses, target our sampling to those 40% of species, and then pull out the viruses that we think might actually someday be a real risk to human health. We can put together the viral discovery pipeline and the risk assessment pipeline. And when we do that, we can reduce the total pool of potential zoonotic viruses by 92%. And we can take that really small subset that we have at the end. We can take the highest risk viruses to a lab, and we can do experiments that actually identify and confirm zoonotic viruses before the first human case. And this is something that we have never really actually achieved as a systemic pipeline in this field. And again, it might seem like I'm about to say that we can use this to prevent them from emerging. The thing is, I don't actually believe that. And I don't think you should either, because knowing about a virus doesn't actually inherently make us more prepared. And again, this is something we can show with really simple data. Maybe the best example of this is influenza, right? Influenza was first isolated in 1933. And since then, we have had 4 pandemics of influenza. It's not for a lack of research effort. We know more about the ecology and evolution of these viruses than probably any other pandemic threat. And we've sounded the alarm probably more than any other threat. But that knowledge doesn't mean we can stop pandemics at the source. Instead, we apply that knowledge to the real tried and true solutions that work for pandemic preparedness: globally coordinated surveillance, vaccine and therapeutic development, and legal and governance frameworks that make us prepared, that operationalize what a pandemic response will look like. We know there will be another flu pandemic, and so we try to be ready for it. We have to start applying that model to the global virome, because today we live in a world that is 1 degree warmer than pre-industrial temperatures. We live in a world that, because of climate change, no matter how much you cut down deforestation, no matter if you shut every wildlife market on Earth, we live in a world today with elevated pandemic risk than the generation of researchers before us. And so we have to start preparing for that world, that reality that we are confronted with. So to go back to what we are doing, let's talk about viral discovery through that lens. When we build a list of the bat viruses that could someday make the jump to humans, we're identifying the scope needed for an effective universal coronavirus vaccine. And that's information that can be used by labs testing out candidates, trying to figure out which vaccines or therapeutics need to be stockpiled. We've seen that a little bit in COVID, but the more viruses we know about, the better our scope will be. And the less likely it will be that a beta coronavirus slips through that filter and we're caught unprepared. But I think maybe more importantly, there are better places to put our money. Despite all of the criticism we can make of virus hunting, and I have been a vocal critic of it, our field is probably not going to stop investing in viral discovery research, but we can make it much cheaper. The money that we save by making viral discovery more efficient can go to more important tasks like surveillance, capacity building, or universal health coverage. Those are the things that actually connect our work to preventing the next pandemic. So on that note, I want to stop, take a step back, and say just a little bit more about where we're headed. Today, I've only talked about maybe half of our data synthesis projects, and our broader goal as a consortium is to be the hub for open data in this field. We want to build data sets that people come back to again and again when they're trying to understand how viruses work or what the next pandemic might look like. That falls into a much broader pipeline of work that we're doing as part of our 10 core priorities, which include ideas like adding more microbiology to these models, or trying to prepare for the next arbovirus epidemic. We're used to the arboviruses going global really suddenly, like chikungunya or Zika, and we know that will happen again. And we're still not sufficiently prepared for that. Even just broadly in the work that we do, we try to build platforms and spread sort of the gospel of open science and make it easier for the people who come after us to do that work. And so I hope that you'll read about it in some of the papers that we've published, in some of our preprints. We try to preprint everything that we do that has any new data or analytical component. There's a ton that will be going up over the next few months. And I hope that you'll be part of that journey with us. The one thing about this consortium I never say early enough in talks is people join our consortium by pitching us a project. If you have an idea, come to us and say, hey, I think one thing you could do with these data is this, and we want to work with you on that. If you want to know more, get in contact with us. I'll do a Q&A. You can talk to me after the talk, or you can find us online at viralemergence.org. You can find us username Viral Emergence on Twitter, on GitHub. All of our code and data are public when our work comes out. And with that, I want to say thank you to, first of all, to my team for putting this together. This is, again, so much bigger than any of us could do on their own. And thank you to all of the wonderful groups that make this work possible, both through collaborative support and financial support. It really makes our world turn. So with that, I want to say thank you for coming, and I'm happy to take any questions. And I'll stop screen sharing.
That was wonderful. Thank you so much, Colin. I see we have a couple of questions in the Q&A. And if folks have other questions, as you sit with it and as we go through these questions, please pose them through the Q&A box. So it looks like the first one we have is from Nilani, and they ask, great talk, thank you. Have you done model validation using a number of different viral genomes to see how well the network model performs to predict the chance that a newly detected virus could infect humans? Does the model perform better for certain types of viruses?
That is a great question. The answer to this is one of the weirdest findings that we're currently grappling with. This is not something I have a fully coherent answer for. This is something that I stare at my ceiling at night and I try to make sense of it. So yes, our network models inherently make predictions about zoonotic risk even before we put them into another model. And we can pull those out and sort of contrast them with the genomic model and see what happens. With the genomic model, there's this sort of test training split. It performs really well on test data. For machine learning buffs in the audience, you get an AUC of between like 0.7 and 0.9. It's a good model. The zoonotic predictions that are actually embedded in the network, when you validate them on the training data, have an AUC of about 0.51, which roughly translates into they are not better than random. And that's specific to this very one network model, this specific implementation, this specific use case. But it's sort of this interesting contradiction in the work where we don't start to get meaningful answers in validation until we start to put in the data that makes good predictions, right? This is sort of similar to the beta coronavirus work, where once we start to put in those viral genomes, that's when the machine actually sort of starts to turn the gears. All of that is also sort of secondary to another part of this question, which is what does it look like to validate a zoonotic risk model? With our beta coronavirus work, it's very easy to get a validation where if we find a virus in a new host, that's a 1 that was formerly a 0. We can't do infection experiments with humans for obvious reasons. And so what we're trying to do instead is come up with ways now to break down that problem in very small operationalized ways. So can we build models that predict cell entry and then do cell entry experiments with different viruses to see how well they work? Can we do models that predict viral replication based on immune factors? And we're trying to sort of figure out how at each step of that process we can do that real validation feedback between models and experiments. But we're still trying to get there.
Thank you. And our next question from Tanya asks, would it be realistic or valuable to generate network embedding for host genomes too?
This is the question that like makes the light go on in my eyes. Genomes are crazy things, right? They are inherently high dimensional data that has millions of dimensions of latent factors, which to machine learning is like every genome is like the entire social network of Facebook. There's so much information in there that can be extracted and worked with. And where we've been up to this point has been a focus on ideas about genomic composition using amino acids or dinucleotides. So for example, we know that there are immune factors in vertebrates that target the GC dinucleotide viruses. And they use that to identify non-self genetic material because vertebrate genomes have a very low GC composition. And so viruses that want to adapt to vertebrates try to match that low GC composition. So that's something that we can put in a model, and we can use that to predict, okay, if it's a vertebrate virus, it probably has a lower GC composition. But even though that's a great way of summarizing the genome, it's collapsing the whole thing down into variables that describe the entire genome at once. And so what you lose is the sort of sequential nature of the data. What you want to do is think about a genome not as sort of a word cloud, but as an entire book where all of the words are in an order that tell a meaning. And so embedding is one of the ways that we can do that. There are a few different models out there that sort of try to do some variation on embedding or deep learning to turn genomes into features. There's a great one called Vorpal that we're starting to work with. One of the best named models I've seen. And we're trying to get to a place where we sort of know how to do this, but it is really early stages stuff, because it's huge data, and it is a massive lift to try to turn those genomes into those predictives. So yes, and we're trying to figure out how to do it.
Our next question comes from Emily, and they ask, thank you for an interesting lecture. Are your models being used yet for the development of pan or universal broadly protective vaccines? If so, what diseases, flu, coronavirus, et cetera, and what sort of collaborative work has been done so far?
Not yet, to the extent of my knowledge. We would love to work with folks. I think there is a lot of infrastructure that could be built for that. One of the difficult lessons of all of this work has been that viral taxonomy is as bad as virologists say it is. You get warned when you're coming into this field. Virologists sit you down and they're like, listen, there's going to be a point when someone tries to talk to you about the new virus taxonomy, and what you got to do is you got to just not look. You got to not make eye contact. And they are not kidding. And all of that affects how we think about a problem. Like, what would we hand vaccine developers if we're talking about SARS-related viruses, right? Because we have all of these genomes, and a lot of them are unclassified. A lot of them don't necessarily have a name. So we're sort of 3 steps before that right now, thinking about like, okay, we're going to go discover some new viruses. We're going to build models that help us do that. How do we keep track of that as an organization in a way that works for us, but also works as an open science resource? And hopefully, if we do that correctly, and we have a lot of brain power coming from folks like, we work with NCBI on this. They are amazing people. They know more about both computers and viruses than I will ever know about anything. And like, we're getting to some answers that I think will support that, but we're not there yet.
Thank you. Jessica asks, Richard Hatchett recently proposed that CEPI should develop a vaccine library against potential pandemic threats, presumably building on the kind of work that you lay out here, combined with preclinical animal studies. Do you think that would help bridge the gap between prediction and prevention, or at least preparation?
Absolutely. I have been very critical of a lot of viral discovery efforts over the last few years, and something that I've never actually found a way to sort of be crotchety and cynical about is vaccine development. I think there are real sort of ethical questions about where we invest our time in terms of vaccine development that parallel a lot of the problems we have on the more applied ends of global health. So in the same way as it might be suspect or unproductive or not match everyone's priorities to be looking for new pandemic threats when we're still not prepared for the ones we know about, I think there's a reasonable case that if we overinvest in a vaccine library for potential threats before we have universal coverage to ensure people have equitable access to those vaccines, or before we deal with vaccine hesitancy as a global issue, we're kind of putting the cart before the horse. And something that we try to write about a lot is this idea that the allure of prevention is always a little bit of a double-edged sword. And we have to be mindful about how we use that, right? We don't want to accidentally sell too much prevention and start to pull the thread of preparedness. And so I think those would be great. I think SHREPI is uniquely positioned to do it because they are focused on these kind of, you know, experimental platforms, these kind of fringe threats. But, you know, at the same time, right, there might be much more value in continuing to work on universal flu vaccines, universal beta coronavirus vaccines, or vaccines for other preventable diseases. Like, you know, a great example of this is we're still struggling to get a good tetravalent dengue vaccine. Like that is at the heart of not just outbreak preparedness, but also a lot of climate adaptation questions. Is like, can we get vaccination to be a solution on the table for dengue at scale? So yes, and also global health is complicated, and I don't have all the answers. But yeah, hopefully.
Yeah, we have a few more minutes, so folks have any lingering last minute questions, feel free to ask now. And in the meantime, I'll ask, so I feel like, and apologies because I'm less familiar in the field, but the obvious jump, you know, the viral research consortium and like everything, people think like, oh, virus vaccine. And so where do you see your work lining up in other realms of like global health prevention and everything?
The big one that's on my mind lately is climate change. I think there is this thing that we do every time we have a big outbreak where we try to solve and in the process sort of try to securitize one very specific threat. So for example, in this outbreak, people have been really focused on wildlife trade, right? But you know, there was a study that came out a few years ago that looked at hundreds of viruses and found that only 6 of them have ever been linked to the wildlife trade. And only 2 viruses have ever actually done the sort of jump from a wild animal to a new animal to humans. It was SARS in 2003 and monkeypox in 2003. Both of those happened 20 years ago. And so there's sort of this analogy that I like to use where it's like someone's broken into your home. And they've nicked your laptop, they've absconded with your TV, and you get really hyper focused on making sure the window they came in through is locked to the point of not thinking about your front door anymore. And to me, climate change is the front door. We want to develop better tools that help us understand climate impacts in real time. And we want to start framing some of this work in the lens of climate adaptation because that work isn't really there yet. There's a couple other questions in the chat, so I might cut that short just to make sure I answer them.
Yeah, thank you. So Thomas asks, how do you account for sampling biases / missing data in your machine learning models? Aren't machine learning predictions sensitive to unrepresented data?
They absolutely are. That is something that we are grappling with all the time. What we are trying to do, so a good machine learning algorithm is designed to deal with that. But what we are trying to do is come up with sort of little tests and little rubrics that actually help us identify that when it happens. So for example, one of my favorite things that we've learned in this process is that blue tongue virus is sort of the most connected virus in the network other than human viruses. And so anytime you run a network model that overfits and overuses the degree distribution to predict, it will almost always have blue tongue virus in the top like 5 to 10 predicted zoonotic viruses. When you fix the overfitting and when you actually sort of change how you're approaching the problem, that doesn't happen. And so what we have now is a little test that we can keep in our pocket where if we run a network model and we get blue tongue as a human virus, we say, okay, this is sampling virus. It's missing data. It's all of those sort of worst evils of machine learning being recapitulated, and that lets us screen our own work before we ever put it into practice. So we're trying to build a little sort of toolkit of those that make us just a little bit smarter.
Wonderful, thank you. So we are at time, so we'll end there. Thank you to everyone for joining us today, and thank you so much, Dr. Carlson, for sharing your work and being a pioneer in this field. It's really important work, and hopefully folks in the audience have been inspired. Maybe we'll reach out with questions or potential projects, and a recording of this will be sent to everyone once it's up. And again, thank you to everyone for joining us today. Thank you, Dr. Carlson, and yeah, happy Friday. Have a wonderful end of your week.
See you folks, and thanks Siobhan for moderating.